Hi all, Dr. Clark here again. Um, Today we're going to continue with mitosis, but now we kind of introduced the cell cycle, chromosomes. Now we're going to talk about the actual nucleus dividing, so what is actually truly called mitosis. Here you can see kind of a diagram of that. This would be a cell prior to entering the M phase. So it could be in interphase, it could be in the beginning of Mphase, but it's you can't tell that the nucleus is dividing. You can tell here that the nucleus is stretching out. The chromosomes are going to be separated. Eventually, that is going to stretch even more and eventually pinch off, cause a cleavage furrow, and then you get two cells. So we're going to go through this entire process in a lot more detail to show you what the chromosomes are doing, how they get separated, and then how does the cell go through cytokinesis. Alright, so first off, if we start with interphase, and you remember interphase is kind of three phases: the G1 phase, the S phase, and the G2 phase. The G1 is the growth phase, and that's normal cell activity. The S phase is what we call the synthesis phase. That's where DNA is going to be copied. And the G2 phase is going to be preparation for the M phase, for mitosis or meiosis. In this case, we're talking about mitosis. So we're talking about somatic cells. All cells except for reproductive cells, germline cells. So G2 phase, it's preparation, meaning that mitochondria or chloroplasts or different types of organelles are probably going to be replicated, at least enough for the new cell to function. Cytoplasm is going to be increased. The cell size is going to be increased enough to support cellular division. And then we're going to move into the M phase. So again, just kind of overview. Chromosomes are duplicated. We already talked about this before. They don't become visible until they go through the condensing process. And that's at the end of the G2 phase and into the M phase. So remember, when I say these phases, these are continuous processes. They're not distinct processes. It doesn't go G1 and then jump straight to S and then straight to G2 and then straight to Prophase. This is all kind of a continuous process. There's, I don't want to call them subphases, but there's intermediate steps in between. So at the end of G2 phase, you're going to get some condensing of chromosomes, and then into prophase, or the first phase of the M phase, you're going to get further condensing of those chromosomes. And we already talked about this when we were talking about chromosomes. Remember that you get the copying of a chromosome in the S phase, and then they bind together with a centromere, so you get sister chromatids, and they're called sister chromatids until they separate, and then they're called chromosomes. And I know the terminology can be a little bit difficult. Just remember that a sister chromatid is just a copy of a chromosome, and they're bound together with centromeres. And that centromere holds them together, but also, like I said before, you get these cohesion forces, so the attraction between the two sister chromatids, they're attracted together, or attracted to each other. And this will play a much bigger role when we talk about meiosis, because that attraction allows for something called crossing over to occur. Okay, and we'll come back to that when we talk about meiosis. Alright, so after interphase, we enter the M phase, or mitosis. And like I said before, it's a continuous process. We divide it out to kind of 4 distinct stages. It depends on who's giving the lecture, what book you're reading or whatnot. really. Sometimes it's divided out to kind of 8 phases, and these would be things like pro-metaphase, meaning, before metaphase, or pro-anaphase or pro-telophase, which means these like sub-steps in between the 4 phases. But for the purpose of this course, we're really going to concentrate on 4 main phases: prophase, metaphase, anaphase, and telophase. Now, like I said before, there are sub-steps that you could put between these and after them, because this is just a continuous process, and it's not jumping from one stage to the next really fast. Some of these processes take a little bit of time for them to occur. And you'll find cells, and I'll show you this when I show you cells in going through mitosis. You'll find cells that are halfway in between prophase and metaphase. And we have steps for this, especially if you're a biologist that studies cell division. Then maybe you're interested in that step in between, because there's some interesting things that are going on in those sub-steps. But for the purpose of this course, again, 4 phases to mitosis. Alright, so let's talk about each one of these phases. Prophase. Prophase is the beginning of mitosis. It's after interphase, so it's after the chromosomes have already been duplicated. You're going to get some condensing of the chromosomes, or further condensing of the chromosomes. So this is really the point at which you can see chromosomes. So when cells are in prophase, that first phase of mitosis or meiosis, we'll get to that, this is the point where you can start to see chromosomes. And when I say start to see them, I mean under 1,000 times your eyesight, you can start to see them. Or maybe you can see them at 400 times your eyesight, but they're not to the naked eye. You're not going to be able to see chromosomes, but nonetheless, this is the point. So once the chromosomes start to condense, I talked about this before, this is a process by which the chromosomes can protect the DNA. So by wrapping it around histones and making nucleosomes and scaffolding proteins, and then condensing it all into that typical X shape, that allows for it to be more rigid and less subject to breakage. And that's because the nuclear envelope goes away. So the nuclear envelope starts to disappear, disintegrate, because you need to allow the chromosomes to come out of the nucleus. This is the only point in time that chromosomes, or that DNA, is outside the nucleus. And that's when the nucleus isn't there, when the nuclear envelope disappears. Okay? The nucleolus, the region where ribosomes are constructed and the RNA for ribosomes are constructed, that disappears also. I don't want to say there's no purpose for it, but it's not needed in nuclear division. If you're talking about animal cells, centrioles start to appear and move to the poles. And so remember, centrioles only occur in animal cells. So if it's not in animal cell, and we're talking about plant cells or other organisms, fungi, protista, these cells, or these centrioles don't exist, but still you get these vesicle packages that allow for the network proteins to form, which we call microtubules. That's the type of protein. They start to form spindle apparatuses, and I'll show you some pictures in a second. I know it can get confusing and hard to imagine these kind of things, but I'll show you in a second. But if you're an animal, you have a specific organelle that allows for these proteins to form. If you're in other organisms, other eukaryotes, you just have vesicles that are normally excreted by the Golgi apparatus or the rough endoplasmic reticulum, and then to the Golgi apparatus, and it allows for the formation of these meiotic spindles. Okay. So those spindles, again, like I said, they're formed from microtubules. We talked about this before. You know, there's three main types of scaffolding proteins. There's three main types of elongated proteins within the cell. Microtubules are one of them, and they're highly important for the process of mitosis and meiosis. So they form these spindle apparatuses, which allows for them to attach to these condensed chromosomes. At the tips of these microtubules, they have a special region called the kinetochore. And the kinetochore is a disc protein that allows for it to attach to the centromere of the chromosome. All right, so we'll come back to that, and I'll show you this in further detail. Okay. But moving on to metaphase. Metaphase is, the next step after prophase and after the chromosomes have already been attached to the microtubules. So, like between prophase and metaphase, all the chromosomes become attached to microtubules. And moving into metaphase, the chromosomes that are attached, all of them, start lining up in the center of the cell. Now, the center of the cell and how they line up has to do with these spindle apparatuses that are attached to these chromosomes, and they're jockeying the chromosome back and forth, but they line up nearly center of the cell. And like I said before in a previous lecture, that center of the cell can be named all kinds of things. Equatorial plane, the metaphase plate, the equator, the center of the cell. I mean, there's all kinds of names for it, but really it's an imaginary line. It's nearly center, and that's where they're lining up. Anaphase, those chromosomes are all lined up in the center, but now the centromere that's holding the sister chromatids together is broken. Okay? So the sister chromatids start to move apart. Okay? And they're moving towards the poles. And so there is enzymes that help with the breakage of that cohesion and connection to the kinetochore. We're not really going to talk about all the enzymes involved. Just note that no matter what we talk about in general biology, if it's a process that needs to have things move, needs to have things combine, needs to have things break down, it pretty much always involves some type of enzyme. Those microtubules then are holding on to the centromere. So the kinetochore, there's a connection between the centromere, the sister chromatid, and the microtubule. And then the microtubules actually start to dissolve. And what's happening is the chromosome is kind of eating its way back to the poles. So it's dissolving the microtubules, the protein, as it moves back to the poles. Sometimes, descriptions of this say that the microtubules are pulling the chromosomes apart. It's not actually true. The chromosomes are consuming the microtubules and walking their ways back down to the poles. So there's actually no real pulling action. There is a little bit of kind of pushing action by some of the microtubules in anaphase that are not attached to chromosomes. So not all of the microtubules are going to be attached to chromosomes. Some are going to be separating or elongating the cell. Remember, these are just protein fibers that can stretch and build and can help support the cell. So when the cell elongates, it doesn't collapse on itself. So you get these elongation of the cell. And as long as the cell is elongating and the sister chromatids are moving to the poles, you can have telophase that happens. The spindle gets dismantled, so all the microtubules are dissolved. The nucleus or the nuclear envelope starts to reform around these chromosomes, and once the nuclear envelope forms around the chromosomes, those chromosomes can start to disassociate with the histones. So they can start unraveling, and it can go back into long strands of chromatin. And so they uncondense, and then of course the nucleolus is going to reappear because you're moving back into a normal cell, one that's not dividing. Alright, so let's check this out kind of in a picture format, a little bit easier to tell. So remember, interphase consists of the G1, the S, the G2 phase. And so you're going to get normal cell growth, you're going to get the copying of DNA, you're going to get the copying of organelles, you're going to get the enlargement of the cell, and you're going to get the condensing, at least the beginning of chromosomes being condensed. Moving into prophase, they're further condensed, completely condensed, okay? And you can see them in their normal, what we would consider, if we're talking about at least mammals, their normal X shape, which is two sister chromatids bound together by a centromere. You can also, in this case, this is an animal cell, you can also see these centrioles that are coming around to the poles, meiotic spindles coming out of the centrioles and going towards the chromosomes that are occurring inside the center of the cell, or close to the center of the cell. You can see this faint line of the nucleus still here, and even in this picture you can see the faint line of the nucleus. That nuclear envelope will eventually start to deteriorate, and before prophase goes to metaphase, it will be deteriorated, so there's no more nuclear envelope by the time you get into metaphase. Metaphase, you can see all those chromosomes are lined up on the center of the cell, the equatorial plane, the metaphase plate. They're also connected to these microtubules. Now you can see here in this photograph or this diagram that you got a bunch of microtubules that are not attached to chromosomes. And again, like I said before, their job is to really provide support but also help with the elongation of the cell as we're moving from metaphase into anaphase. So you can see that in anaphase, the cell itself is elongating, but also the sister chromatids are being separated and walking themselves back to the pole. And so here you can see this is an actual photograph of it occurring. You can see the chromosomes are in purple, and then kind of that pinkish color is going to be that meiotic spindle, and they're walking them back. Now you can see the meiotic spindle, maybe you can see it in between here. You still have meiotic spindle that is helping with the elongation of the cell. Telophase, those sister chromatids have now made it to the poles, and a new nuclear envelope is starting to form around it. At this point, these are called chromosomes. They're no longer called sister chromatids. Remember that denotion is really just telling you that it's a copy of a chromosome. And you can start to see here, this is an animal cell, so you start to see this cleavage furrow start to form, and it's going to pinch off and move into cytokinesis. Now remember, cytokinesis is not part of mitosis. Cytokinesis is its own phase, and that's really just the division of the cytoplasm, the division of organelles, and the reformation of plasma membrane in this case because it's an animal cell, but also the formation, if this was a plant cell, also the formation of a cell wall, or what we call cell plate at first, to separate the two nuclei. So cytokinesis will follow telophase. Not always. Sometimes you get telophase occurring and you get two nuclei inside the cell and it never made it to cytokinesis. It stopped there. This is a mutated cell, and normally we'll go through something like apoptosis, which is programmed cell death, because there's something wrong with that cell. Sometimes they stay, and you know, there's slides you know, that I have in lab that show 2 nuclei in the same cell. It happens, but nonetheless, mitosis already occurred because you have 2 nuclei. Cytokinesis then has to occur, or the C phase then has to occur to separate the 2. All right. So back to cytokinesis a little bit. It occurs at the end of mitosis and at the end of meiosis, and it is the process of dividing the cytoplasm in half and the division of, or the separation of, organelles. Now, when we talk about mitosis, we say that mitosis is a identical copy of the cell. Now, when I say that, what I mean by that is it's an identical genetic copy of the cell. The 2 cells after cytokinesis they could be identical, but often they're not identical. So maybe, you know, the cell on the right has more mitochondria than the cell on the left. This happens. This happens often. Okay? So the cells are not completely identical, except for the genetics inside their nucleus. That is identical, unless there was a mutation that occurred, and then that's a different story. But through normal cell division, so you got a little cut and you need to repair that, normal cell division, genetically speaking, those cells are identical. Okay? Maybe not when we talk about the overall composition of the cell, like how many organelles they have, how many ribosomes they have, the size of their endoplasmic reticulum. You know, that might all differ, right? But inside the nucleus, their DNA is identical. Okay, so again, depending on what type of cell we're talking about, cytokinesis happens in a kind of different form. If we're talking about animal cells, cytokinesis has to do with the actin filaments. Remember, we were talking about those. Those are the protein filaments that make up one of the 3 protein filaments that are common inside the cell. Actin filaments have the ability to contract, and when they do so, they can contract in the center of the cell, and they can form a cleavage furrow. So it forms kind of an indentation, and as that indentation keeps occurring and the cell keeps elongating, eventually the cell will pinch off in the center, causing what we call 2 daughter cells. In plants, because plants have a rigid cell wall, all plant cells are attached to the plant cell next to them. So what happens with plants is, instead of, you know, pinching off and forming 2 cells, they just form a new barrier between the 2 nuclei. And then the cells can elongate as they grow. And that new barrier is called a cell plate, and eventually it becomes a cell wall when more cellulose is laid down. Now, this is a result of vesicles sending cellulose to the region, to the center line, to form little vesicle connections, and eventually it'll form a cell plate in between. And I have a picture of it here showing that. Right? So here is the animal cell pinching off through the actin, condensing of the actin, or movement of the actin fibers to pinch off and cause a cleavage furrow, and eventually 2 cells would originate from this. And here you can see vesicles. These vesicles are normally coming from either the smooth endoplasmic reticulum, because a lot of these are going to be lipid-based with some carbohydrates. So some of these vesicles might be coming from the Golgi apparatus also, and they line up along the center of the cell, or basically halfway in between the 2 nuclei, to form this cell plate that eventually becomes a cell wall. So eventually, over time, new layers of cellulose will be laid down, and this will become a rigid cell wall that separates these 2 cells.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, so that, in a nutshell, is mitosis and at least a piece of cytokinesis. The next thing that we're going to talk about is programmed cell death, or in some aspects, programmed cell cycle. So how is it that we regulate when cells replicate themselves and when cells have had enough? So how damaged, or if the DNA in the cell is damaged, or if the cell's really old, what happens to that cell? How do we get that cell to dissolve itself or consume itself, which we call apoptosis, or programmed cell death, and how does this work? Okay, so we'll come back to that next time.